All right, so we're finally here, step 3 of cellular respiration. So we're going to be talking about how the electron transport chain works, how we're going to be using these electrons that we have stored, I guess you could say, in these electron carriers, or being carried by these molecules, NADH and FADH2, and how we're going to use them to power the process of making lots of ATP in oxidative phosphorylation. Okay, so here we have a picture of the electron transport chain, and this is where understanding the spaces and membranes of the mitochondria becomes very important, very, very important. So let me orient you to the diagram. They show you a little mitochondria over here on the left. This membrane, this is a lovely phospholipid bilayer with some proteins embedded, some transmembrane proteins, and then some protein electron carriers. This is the inner membrane. And then this would be the intermembrane space. They nicely label that for us. And then up here, where it's not labeled, would be the outer membrane. So the space down here is the mitochondrial matrix. Okay. So remember, the citric acid cycle, the Krebs cycle, was going on in the matrix, turning and turning out many, many NADH molecules and FADH2. And these little blue spheres represent our electrons. The electrons this single molecule of NADH is carrying. Okay. All right. Now, before we get started on the process, let me also orient you to these proteins and these electron carriers of the electron transport chain. So the electron transport chain is located in the inner membrane of the mitochondria. It consists of 3 proteins that span the inner membrane. You can see them here. And 2 protein complexes that carry electrons from one protein to the next. Okay. So electrons are going to start up here at this protein of the electron transport chain. And then as the yellow arrow indicates, the electron carrier is going to carry it to this next protein of the electron transport chain. Another electron carrier will carry it from the second protein of the electron transport chain to the third and final protein of the electron transport chain. All right. So the electrons, if it's being carried by if they are being carried by NADH, will be dropped off at the first protein of the electron transport chain. If they're being carried by FADH2, they're a little bit lower energy. They're going to be dropped off here at this first electron carrier. And when the electrons are dropped off, they are losing electrons. The molecule NADH is losing electrons. It's going to be oxidized to NAD+. So then that NAD+ can be used, again, it can be recycled by the cellular respiration machinery to go through another round of glycolysis or used in the citric acid cycle. So it's not gone to waste. It's not put to waste. And then the same with the FADH2. It is oxidized to FAD. All right. So let's go to looking at this process a little bit more detailed with some directions here at the very bottom of the screen. So again, those electron carriers dropping off their electrons at the electron transport chain. And when the electrons come in contact with the proteins, these membrane proteins that span, these transmembrane proteins, those electrons, remember we said they're energetic. They give these proteins energy to pump protons, H+, hydrogen ions they're also called, from the matrix, this is important, from the matrix to the intermembrane space. Okay? And then the electrons are carried by the electron carrier to the next protein, if we're going with electrons from NADH, and if they're coming from FADH2, same idea. They give this protein energy to pump protons through the channel in the protein to the intermembrane space. And then the electrons are carried from the second protein to the final protein, and the electrons give that protein energy to pump protons from the matrix to the intermembrane space. So why do we need energy to do that? Well, as you can see, there's a much higher concentration of hydrogen ions, protons, in the intermembrane space. There's a lower concentration of hydrogen ions in the matrix. So it's kind of like we talked about in the chapter on cell membranes and transport across the membrane. This is going uphill, going from low to high, like rolling up a hill. That would take energy. So the energy comes from these high energy electrons that we have stripped systematically from glucose. And as we know, glucose is transformed into many intermediates, both in glycolysis and in the citric acid cycle. But the original molecule was glucose. So we can say that they are stripped from glucose in the processes of glycolysis, and most notably the process of, or more so I should say, the process of the citric acid cycle, or the Krebs cycle. Okay, well, what is pulling them? What is the driving force pulling them down this electron transport chain? Why are they moving from protein 1, being carried by the electron carrier, moving to protein 2, electron carrier, protein 3? What is the driving force? Well, as you can see, the arrow is pointing to oxygen. And that, my friends, is the main reason you breathe oxygen. That is the only reason. Take a deep breath, and that helps you pull electrons down the electron transport chain, they say, from one protein to the electron carrier, to the next protein, to the next electron carrier, to the next protein, and finally to oxygen. And then those electrons combine with oxygen, and then that combines with hydrogen ions to produce water, one of our major products of cell respiration. Okay? Oxygen being one of the main reactants, water being one of the main products. Okay. So we talked about step 5 here. Hydrogen ions are pumped through these protein channels as the electrons come in contact with them, as they move down the electron transport chain. And while we do that, we are creating a very high concentration of hydrogen ions in the intermembrane space. Those hydrogen ions are positively charged. The fat in this membrane does not like this polar, water-like molecule. So these hydrogen ions cannot easily diffuse through the membrane. And they can't go back through these protein channels either. These protein channels, my friends, are a one-way street. Only one way these protons can be pumped from the matrix to the intermembrane space. There's no going back through these protein channels. So the only way these hydrogen ions have to get back into the matrix, down their concentration gradient, so to say, and to try and equal out across the membrane, is to go through a wonderful enzymatic molecule called an ATP synthase. And the ATP synthase is also located in the inner membrane. And as the hydrogen ions move through the ATP synthase, it kind of turns the ATP synthase like a wheel under a waterfall, I guess you could say. The hydrogen ions being the waterfall and the wheel being the ATP synthase. And it allows that ATP synthase to act as an enzyme and take ADP, adenosine diphosphate, and add a phosphate bond, or add a phosphate, bond it to the other phosphates in ADP, and that's where we make our ATP. Okay? So let's think about this idea of oxidative phosphorylation. What's being oxidized? We already talked about it. NADH is being oxidized. It's losing electrons. What's being phosphorylated? ADP is being phosphorylated. A phosphorus group or a phosphate group, is being added to ADP to make ATP. Another term you might come across, chemiosmosis. Remember, osmosis is the movement of water. So they liken that to hydrogen ions. Hydrogen ions are polar. They're not water, but they're polar. So I can see where the osmosis part comes. And they're moving across a membrane. And they're chemicals moving across a membrane, I guess you could say, ions. So chemiosmosis, the movement of the hydrogen ions across that inner membrane through the ATP synthase and turning that turbine of the ATP synthase and allowing it to make ATP. All right, so one last thing we want to talk about before we end this review on cellular respiration is the idea of toxins that could interrupt this process. One of them in particular shown here, rotenone, is actually a pesticide. It's not used commercially, but it is used currently to get rid of invasive species, fish species, in certain bodies of water. And it actually prevents the transfer of the electrons to the electron carrier, this first electron carrier. So let's think about what that would result in. So if we can't transfer those electrons to the electron carrier, the only proton pump that would be actually working would be this first proton pump. So we would only be pumping protons across the membrane through this one pump. These other proton pumps would not be functioning. So we'd only have about 1/3 as many hydrogen ions in the inner membrane space. And then only 1/3 of the hydrogen ions moving through the ATP synthase. So we'd only create about 1/3 of the ATP that we need. And that is not good. So this would affect the amount of ATP that the organism could produce. And like I had said, ATP is the currency of the cell. You don't have ATP, you are not going to do well. You only have 1/3 of your ATP, you're not going to live very long. So this is a way to, again, that they use currently to deplete or to get rid of invasive fish species in certain bodies of water. This particular pesticide has been linked to Parkinson's disease. So it is kind of on a chopping block right now in terms of its continued use in publicly accessible areas and bodies of water. But the other poisons here are shown, and I'd like you to think about, how each of these poisons would affect the production of ATP and its effect on the organism. Okay. So hopefully this overview has given you a better understanding of the process of cell respiration. And please feel free to contact me with any questions that you have.